Okay, let's have a class. And we will talk about like 55 minutes today. We'll end up the class like 12 o'clock. We will talk about is applied microbiology second section, which is pathogen control during the poultry processing. Because this slide has lots of pictures, so it's not available to upload PDF, PPT into the eCampus. So I can only do PDF. Okay, but some of you like that. So what we're going to talk about is the pathogen control during the poultry processing. This is an overview picture of broiler production in the United States. The survey which is done by USDA and ASS in 2015, which means the data is from 2014. So you can see the majority of the broiler production in the United States is in the south part. Georgia, Alabama, Mississippi, Arkansas, Texas, North Carolina. This is the reason all those places have a good poultry science program. For example, University of Georgia have a very good poultry program. Alabama, Auburn University have a poultry science program. Mississippi State University, University of Arkansas, Texas AEM, and North Carolina State University. Their poultry science program is very important for their local community. So if you are interested for your graduate study, if you want to do a poultry research, those are the places you should go. Now, part of the reason I always make a joke is because the birds usually, the intestinal temperature is like 42 degrees Celsius. So in this warm temperature, usually favors there to grow, to survive. Although it's not really corresponded, but it's really the reason for that. So you can see Georgia, 1,324 million head. It's a huge. Alabama is more than 1,000 million heads. Okay, so that's we also say the 6 universities in the south, they have a poultry science program, which means they have a department of poultry science. Let's look at it here all the way above. Kentucky have 308 million head. Kentucky used to want to do some poultry, become a poultry state, but the problem is that they shut down that program. The reason is the waste is very difficult to control. Virginia have some, usually across the border with Virginia and West Virginia, in the Hardy County, in the Eastern Panhandle, it's a big place for the poultry. If you look at this map, it's surprisingly West Virginia generates 95.3 million head every year. It's ranked about like 13 or 14 in the United States. And we don't have a big agriculture here, you know that. We are historically is mining, the mining state. But agriculture, the poultry, is one of that, and it is very important. That's why we want to talk about it. And most of our poultry processors are very small poultry producers. Okay, so that's why we want to talk about it. It's important. What are the major pathogen concerns for the poultry processing? It is Campylobacter and Salmonella, but I want to be cautious to you. Not only for that, or other pathogens also could be shown up. So for example, Listeria monocytogenes, nobody really cares about. Recently they showed during the cage, in the cage of the poultry, when they're raising that, they find a high frequency of Listeria monocytogenes. When the final meat products generate, during the cooking processing, Clostridium botulinum could be a concern because it's an endospore pathogen. And E. coli 01578H7 does not report it too much, but I tell you when seeing the poultry meat, sometimes it also causes contamination. So, just want to let you know, although we keep talking about Salmonella and Campylobacter, but all the others could be still a concern in the near future. That's why the last thing here we mentioned is the emergent organisms. So let's talk about the Campylobacter first. Okay, Campylobacter, very important. This is Campylobacter. Campylobacter. This is a gram-negative lozenge-shaped bacteria. And this lodge is very interesting. It's a curve. Looks like a vibro. Campylobacter, the major characteristics is they grow usually 42 degrees Celsius. It needs microaerophilic environment. It's very interesting, which means their oxygen somehow like 5%, carbon dioxide 15%, and the rest of them are nitrogen. It is very ironic. This pathogen is difficult to grow in the lab. We need a jar, a special microaerophilic jar, or even a microaerophilic chamber. And the microaerophilic chamber sometimes $25,000. It's expensive. However, it is a big concern in the poultry industry. Because it's difficult to grow in the lab, we cannot do much research about that. However, it's a big concern. So it's very ironic. But we find out some way to do with the development of the science. So that's why tons of research start to do the Campylobacter. But I tell you, it's very difficult to grow. I'll give you a very easy example. E. coli you can grow in neutron broth. If you pick colony from Campylobacter plates to grow in the neutral broth, it does not work. It won't even grow. And let's say E. coli, you can do streak plating onto neutral agar and tropic soil agar, those supported medium. Campylobacter, you can do that. It doesn't even grow. If you streak onto neutral broth, neutral agar, even you put into microaerophilic chamber, it still doesn't work. It has a special media sometimes need using vitamin K as a supplement. You need the blood there. You need other ingredients. All those needs to be added onto a modified chocolate agar. So we all call Campylobacter is a typical fastidious bacteria. Okay? So here you can see it's relatively fragile, sensitive to environmental, requires 3 to 5% oxygen, 2 to 10% carbon dioxide for the optimal growth. And the Campylobacter, they are not only for the poultry originally, it's existing a wide range of the domestic animals and the wild animals. And they could be survived relatively longer time, 2 to 4 weeks. And the temperature could be low as 40 degrees Fahrenheit. Which means if the poultry meat you stored in the refrigerated temperature, this bacteria will not be killed. It's going to stay there. It could be potentially grow even. But the good thing is that it's different from E. coli 01578H7. E. coli 01578H7, the infectious dose is very low. And 10 cells will kill people. This infectious dose is 500 to 10,000 cells. So that's a good thing for it. Second one, Salmonella, we already talk about that all the time. Salmonella, very similar to E. coli. It is gram-negative, it is a large shape, and it's a facultative. And no endospore forming, so no endospore. No spore forming. And this is widely existing in the world, and not only for chicken, even for produce, it's a big risk. And it can grow very well at body temperature, so we call it a mesovirus. And the Campylobacter, you can grow basically in all the supporting media. So it's not a fastidious bacteria. It's easy to do research about that. And you can see here as optimal temperature 35, 37 degrees Celsius. And the maximum 45, 47 degrees Celsius. This is the key here I want to mention when we talk about the processing. They can grow 45 to 47 degrees Celsius. This is going to be the key. Okay, remember, it's could grow. They could grow as low as pH 4.0 and up to pH 9.0. So a little bit acidified. Now, of course, the optimal pH is still the neutral. That is basically universal for most of the foodborne pathogens. This is a survey in 2005. It's a top 30 serotypes for the Salmonella. The number one, Salmonella typhimurium. So you can see there's close to 7,000 of the cases happens. Then followed by Epideritis, Newport, Hindenburg, and Montevideo, all these, which is usually we call the top 5. And let's go down to see Infantitis, Thompson, Limited. And usually for the poultry meat products, we care about this typhimurium, the first one. For the eggs, we rarely think about enteritis is a big risk for the eggs. Okay, this is something I would love to emphasize. It is different from E. coli OM57H7 we talk about in the lab, which is zero tolerance for ground beef or non-intact beef. Salmonella and the Campylobacter is not zero tolerance. They require you is below a statistic survey sampling plan results. This is a new performance standards for the poultry carcasses start from January 2015. Salmonella, as long as it's below 7.5% positive rate, you're good. Campylobacter, less than 10.4%, you're good. That means when you buy a poultry carcass from supermarkets, from farmers market, there is no restriction for zero tolerance, which means possibly you could get salmonella. That's the key. That's why the chicken meat products you have to cook well done. But that makes sense when you go to restaurant order chicken steaks. You never see I want a rare, I want a raw chicken. And if when you go to the restaurant, if you order something, you see the chicken is raw, please ask them to recook it. Because Salmonella and the Campylobacter have a certain percentage in a certain type of the products. It's not really zero tolerance. Okay, so this is something we have to be emphasized about that. Another thing I also want to tell you, started from just July last year, FSIS has a new regulation for breaded chicken. Because the breaded chicken, who is the big target, a big consumer? These kids, breaded chicken nuggets. And there are several outbreaks happens. So the breaded chicken nuggets, FSIS, requires zero tolerance for salmonella. But that puts lots of pressure for the small poultry processors, which means there's lots of the interventions they have to do. Because breaded chicken, we call it non-ready to eat meat products. They usually just flashing cooked in or deep oil. As long as the breaded is brownished, we call it breading, then they can be frozen and then they sell to the markets. And basically at home you need to use an oven to recook it. But right now they have a regulation, even those breaded chicken in the packaged sample, you have to be zero tolerance for salmonella. It's just brand new, okay? So in other words, Salmonella Campylobacter more or less will exist in the poultry carcasses. So that's a new performance standards. Okay, what is the standard broad processing in the United States? The grow out transport, hanging there, immobilization, we call it bleeding out, the terminology called exaggeration. Then we're going to go into a scattering processing. That's a major point for cross-contamination. And the pickling, evisceration followed by, then we do a chilling. Between evisceration and the chilling in the United States, if you're in a big company, you usually hand the birds back to do the antibiotic washing. We call inside-outside birds wash. Then after chilling, we do an aging, which means a vacuum package in a certain temperature, lower temperature for a certain time, and we do the sorting, and finally we do the processing, and we do the packaging. And labeling, that's also important. So we want to talk about several steps, which is important for the pathogen control, since this is the microbiology class. First of all, most of the large companies, they do, before scattering, they do a pre-scattered bird's brush. So why? You know the birds, it just takes out just a freshly exaggerated. They have more or less some organic loads on the surfaces. So we want to lowering the organic loads, especially the fecal contamination. We do the pre-scattering brush, which is a physical removing activity, could also decrease the bacterial load when they go to the scattering processing. Now, the feeds usually feeded by the poultry, and then they will be eaten. More or less during the mashing process, they'll have some of the contamination. Now, the last step, when they go to the chilling processing, there are the organic matters on the surface, there could be bacteria on the surface. Pre-scattered bird's wash could also reduce that amount. So, most of the company, what they do, before they do a scattering, they use brushes to automatically to brush the surface. Remove those dust, debris, and some residual fecal samples. So that's a pre-scattered bird wash. Okay, next step, scattering processing. Scattering processing is a very old process. Basically, you're going to be exposed, the birds go to the hot water. They're easy to do defeather. It is started the 1st century AD, so you can see the picture there. It's a very old procedure. You can read the sentence here. The partridge is scattered with its feathers, and the wild wet feathers are taken off, and the hair seen. So it's coming from the 1st century AD, and it's a long, long time people doing the scattering. And however, we have to emphasize that scattering is a major point for cross-contamination, because they are from the skin surfaces. From a contaminated carcasses will be spread to uncontaminated carcasses. And when we see a scattering procedure, we will really care about flow rate, fast or slow. The direction, is that a countercurrent to the water flow? And we will talk about the importance of that. Is that a multi-stage or is that a passing scattering? And the temperature control is very important. This is what I mentioned about here. Some of the small producers, they want to make sure the processing is easy and comfortable. They are using relatively low temperature of the hot water. So for example, some of them, they do 45, 47 degrees Celsius, even 42 degrees Celsius. That's really a bad choice. Because that low temperature, if you have 100 or 1,000 birds to processing, what you are doing? You are cultivating salmonella. Because salmonella can grow really well and fast at 45, 47 degrees Celsius, much faster than at 35 degrees Celsius. So the joke is saying, if the temperature of the scattering you didn't control very well, it's an expensive inoculation chamber. And of course, the condition of the water where the birds exit is important. It's have to be as clean as possible. And also the condition of the birds when they exit. Is there visible fecal material? If there is, it could be a resource for cross-contamination later. Okay, this is what we're going to do a scattering. Very important in the temperature control. There is basically two types of the scattering processing. Number one, you could use a relatively high temperature for that. You could do like 59 to 64 degrees Celsius, and that could do a little bit short time, around 30 seconds. And if your choice is a relatively soft, we call it a soft scattering, you could use a lower temperature. That's a temperature relatively 51 to 54 degrees Celsius. Now, this is very important. This could be a little bit longer, 60 seconds. That's what we did in our poultry farm. This temperature is very important to control. We said that you cannot do very low hot temperature. Make yourself comfortable. But in opposite, some of the small processors, they are saying, it may be a major step to killing bacteria. How about let's use very hot water? Let's do 70 degrees Celsius hot water. And I tell you one thing, this is the wrong procedure. Why? If you do 70 degrees Celsius scalding, the skin surface of the birds, there is fatty acids and the lipids. You know that, lots of the oil. If you just cooking a skin of a bird's boil, you will see a soap floated with oil. If you do scalding 70 degrees Celsius, it will be doing lipidation of skin surface. Then what happens? Those lipids, those fatty acids will be extracted out of the skin surface, and when they do the final chilling steps, all those will become organic matter and floated on the chilling tank. The organic matter is a big disruption for the efficacy of antimicrobials, especially chlorine water. So we will mention that. That's why this temperature control is very important. Another important thing is we mentioned about the water flow. The water flow has to be countercurrent, which means the birds come in, you're always facing the clean water. However, it is difficult to do. There is some of the surveys showing lots of the extension specialists to go to the south part of the United States, and they talked to the owner of the poultry processing plant. They told them we are doing the countercurrent water flow. However, when you go to see it, usually it's not. Why? Because the design of those water flow is from a food engineering or engineering standpoint. And they do not have really a concept for microbiology, especially the pathogen control. So, not many of the people really do countercurrent water flow. But I tell you, it is important that you have to do the countercurrent, which means every time the birds have to face in the clean water. So that's a scalding. So these are typical examples for the scalding. You can see it. Another thing we also want to mention, scalding procedure. We're using a hot water. Can we add antimicrobials there? Yes, you can, but very limited. First of all those oxidants chemical, you cannot use it. Chlorine dioxide, hydrogen peroxide, please don't use it. Why? Because in a very hot temperature, they will generate off gas. The off gas will make people sick. And also, basically, scalding water is a high amount of water, organic matter. They will decrease the effective concentration of the antimicrobials dramatically. And I will talk about the chlorine real quick. They could decrease 50 parts per million of chlorine water, the concentration, to zero about like a minute or even 30 seconds. So we don't use too much of the antimicrobials. If you want to do only those strong acids, organic, inorganic, or mixture of the acids you can use. Hypochloric acids you could use. But I tell you, with the slaughtering procedure, relatively less people are using antimicrobials. Okay, pickers. Pickers is very important. You hand in there, you're going to see more or less there are some of the fecal sample going to come out. Why? Because you're extending the area of the ceca and the viscera. Very important, the concept here is feeds withdraw. In the United States, our target feed withdrawal time is 10 hours. This feed withdrawal is important. You cannot do it too less time. Why? Your feed is in the intestine. When you do the evisceration, easily to broken the intestinal area, the ceca, the visceral area, what happened? It causes cross-contamination. You cannot feed withdraw too long. If you feed withdraw more than 36 hours, what happened? The intestinal area, the ceca, could become soft. So, feed withdraw 10 hours is usually a standard, and viscera seems like ribbon. This is a standard. When you do the visual inspection, if your viscera looks like a ribbon, that looks good. If you touch it, okay, if you see it's too soft, that's not good, which means the feed withdrawal is too long. If you see it's still lots of the feeds there, that means your feed withdrawal is too short. Okay, so pickers, what happened? Lots of the time you squeeze it. Okay, you make the feeds more, comes out, it's a cross-contamination. And this pickers is actually a significant resource to increase salmonella preference in the Campylobacter, especially for the Campylobacter. Because our research and also previous research also showing Campylobacter is a high number in the ceca. Because during the pickering process it could be expressed. Okay, evisceration. This is usually manually we do it, or some machine we could do it. For very small producers, they just manually do it, same as we did in our poultry farm at WVU. This is a major component for cross-contamination because you are taking out of the intestinal area. If you do not do very well, it could be contaminated the whole carcasses. So this relates to the equipment. If it's not adjusted correctly, your feed withdrawal is not enough. You have a weak, soft intestinal, then this is a big resource for cross-contamination. So what we do? We need